Okay, now we're going to talk about our muscle tissue. Now, our muscle tissue is distinct in that we're going to have contraction of our muscle so that it is going to shorten with force, and that's what allows us to move around. So we will move our entire body, and also contracting our muscles will help us to pump our blood. So we have three different types of muscle tissue. We have skeletal, which was attached to our skeleton. Some are attached to other types of connective tissue, like we do have some skeletal muscle that lies within our subcutaneous tissue. And this muscle type is going to be striated, meaning we have these altering colored bands throughout them, so light, dark, light, dark. And these are voluntary, meaning we can consciously think, I want to move my hand, and we're able to do it. The other type is going to be our cardiac muscle type, and this is, of course, the muscle within our heart. It is striated as well, so we have those different colored bands, and this is involuntary. We are not able to consciously contract our heart just by thinking it. And lastly, we have smooth muscle. This is not striated, and it is involuntary. And we're going to find this type of muscle within tubular structures, so within our digestive system and also within our skin. So this is a nice little chart here that talks about location, our cell type, nucleus, striations, control, ability to contract spontaneously, function, and our special features. So I'm going to discuss this as we go through each type of muscle. First, we have our skeletal muscle. Like we said, these muscles are going to be striated. So here's a great histology slide of what a typical skeletal muscle type of tissue looks like. We can see the difference in these bands, dark band, light band, and so on. These cells are going to be extremely long, and they're cylindrical. And what's unique about skeletal muscle is that they're multinucleated, meaning one cell has multiple nuclei. So we can see 1, 2 just on this muscle cell alone. The function of skeletal muscle is to move the body. It's also under conscious control, so we can move however we want. And location is that it's attached to bone and other connective tissues. Our cardiac muscle tissue is going to be found within our heart. So we still have that striated appearance, but what I want to focus on is how do you tell the difference between cardiac muscle and skeletal muscle for your exam? Well, first, yes, we have that striation, same as skeletal, but the difference is that this cardiac muscle is branched. So it's not long and cylindrical, but you're going to see it branching off to the side like this. One of the other differences that you'll see is that it contains intercalated discs, which will have special junctions that are going to include those gap junctions that we discussed earlier, that it will allow ions to move from one cardiac muscle cell into another so that we can have contraction of the cardiac muscle altogether. And I think I already mentioned that this muscle has striations. So the only place we'll really find cardiac muscle is in the heart. Nope, went a little too fast. Lastly, we have smooth muscle, and these muscle cells are very interesting because they have a little bit of a spindle type of shape to them. They're more tapered at the end of each muscle cell. They also don't have any striations, and that's why they're called smooth muscle cells, because we don't have that alternating light and dark bands here. And they only have one nucleus. That reminds me, for our cardiac muscle, we will only have 1 to 2 nuclei per cell. This is different than our skeletal muscle that is multinucleated. Okay, back to our smooth muscle. Another note here is that that nucleus will also be centrally located. So when you look at this muscle, this smooth muscle in a histology slide, you'll see these pink, they look like fibers. So I don't want you to get this confused with collagen, but they'll have a little bit more of an elongated nucleus right in the center of each of the muscle cells. So you want to kind of pick a nucleus and then try and outline the actual muscle cell. And you'll know that it's smooth muscle. There's no striations. There's no intercalated discs. So this absolutely has to be smooth muscle. And we'll find these in hollow organs. So a lot of our organs in the digestive system, such as the stomach, our small and large intestines, as well as within our blood vessels. All right, that brings us to our nervous tissue, which is our last type of tissue. Our nervous tissue has two major types of cells. We have our neurons or nerve cells, and we also have neuroglia or glial cells, which we'll talk about in a moment. So first, let's discuss these neurons. They have the ability to produce action potentials. That's another word for a nerve impulse being sent through the neuron in order to communicate with either another target organ or another neuron. So our neuron has a cell body, which is where we'll have our nucleus along with majority of our other organelles. It also has an axon, which is this long process over here, and this is the area where our action potential will move away from the cell body and over to the axon terminal or the axon ending here. And I also want you to notice our third structure we're going to learn about, which are the dendrites. And these are cytoplasmic extensions kind of reaching out that will receive impulses from other surrounding neurons out here and can transmit that information along the axon if it's stimulated enough. Now, we also have three major types of neurons. We have multipolar, which you see pictured here, bipolar, and also unipolar. So here is a slide on our multipolar neuron. These are the most common ones throughout the body. We see the dendrites sticking out in our histology slide, its cell body, the nucleus here, and we see an axon extending on out to transmit that nerve impulse or action potential. We'll also see that we have some nuclei of neuroglia. So let me talk about what neuroglia are. Neuroglia are supporting cells for our neurons. So I've described this within my biology course too, but it's almost as if the neuron is the main celebrity. So think Angelina Jolie. She has a team that surrounds her to help support her and nourish her, right? So she probably has a chef, a personal trainer, a makeup artist, a hairstylist, manicurist, did I say stylist, and so on. You know, the list goes on. So they are all working to support Angelina Jolie. Angelina Jolie being the neuron, and the neuroglia being all those different occupations, the hairstyles, manicurists, and so forth. So anyway, that's what we see in this histology slide. The function of our neurons is to transmit that information, that nerve impulse or action potential over to either a organ or to another cell, another nerve cell that is. We'll also see these sheaths that are surrounding the axon, and these are called myelin sheaths. We actually have two different types of neuroglia that can produce these. The one that's pictured here is our Schwann cell, and our Schwann cell is a type of neuroglia that is found in the peripheral nervous system. The neuroglia that creates these myelin sheaths in the central nervous system are called oligodendrocytes. So the areas where we'll find nervous tissue will be in our brain, in our spinal cord, in our peripheral nerves, as well as ganglia, which the definition of ganglia, which is plural, or ganglion, which is singular, is a collection of cell bodies within our peripheral nervous system. So here's a slide on a pseudo unipolar neuron. We said pseudo means fake. So this looks like it's a unipolar neuron cell. So let me go back to show you our unipolar. Okay, so we got the two sides and our cell body is out here. But this one is a fake, it's an imposter. So we see the dendrites extending on out in this direction here, and our cell body is moved on up here, and then we can transmit that information along the axon to the axon terminals. Oh, and I forgot to point out our histology slide here. We have our nucleus of our neuron surrounded by the cell body, which would contain all those other organelles. We also see surrounding each of the cell bodies here are the nuclei of our neuroglia. And here's another image of our neuroglia. Also, just to mention real quickly, they are also called glial cells. It means the same thing. But here are those oligodendrocytes that I was talking about in your central nervous system that create the myelin sheaths. And we also have these beautiful star-looking like glial cells called astrocytes that'll do things such as create the blood-brain barrier. They'll pick up any extra ions that are floating around. And these are just 3 of the neuroglia that I've mentioned out of 6 total. So we'll continue listing the rest of these glial cells in our nervous system lectures in the future. And the last thing we're going to talk about are tissue membranes. I think this is the last thing. So this is a little bit of a review from chapter 1, but we didn't really discuss our mucous membranes. They're going to line our cavities that are open to the outside of the body. They secrete mucus. They contain epithelium with goblet cells, and we know that our goblet cells are the ones that are going to produce and secrete the mucus. Below our epithelium and our goblet cells, we have that basement membrane. And remember, below that, we have the lamina propria, and sometimes it can contain some smooth muscle in there. We'll find these mucous membranes lining the respiratory tract, our digestive tract, and urogenital tract. Next type of membrane that we have is our serous membranes, and this is the one that we discussed in chapter 1. So remember that in our lungs and our heart and also in our abdomen, we have these serous membranes, and we have specific names for them. For our lungs, it would be our pleural serous membranes. For our abdomen, it would be our parietal serous membranes, and then surrounding the heart, we had that pericardium. So all of these serous membranes are made up of simple squamous epithelium, and we give a specific name to it called mesothelium. Below that, we have our basement membrane and then a thin layer of loose connective tissue. These cavities we already mentioned, so I won't go through them again. And the last membrane type that we have are synovial membranes. These are going to line all of our synovial joints, which will be our freely movable joints. So as you move your fingers, your shoulder, your knee joint, your hip, these are all examples of synovial joints. And the synovial membrane here is going to assist in producing our synovial fluid, which is going to be rich in that hyaluronic acid. Oh, I forgot, we're going to talk about tissue damage and inflammation and repair. So we have quite a few slides ahead of us. So let's talk about tissue damage and inflammation first. It is the way that our body is responding to any type of invasion in our body. So usually when there's an invasion from the exterior of our body inward, we're going to have some tissue damage, and our body will respond with our immune response. And the manifestation or the cardinal signs of inflammation that we'll have are redness, heat, swelling, and pain. So those are the 4 cardinal signs that most people know of. They've just added a 5th cardinal sign, which is going to be disturbed function or inability to function within that location. So we'll also have some chemical mediators that'll be released when we have any tissue damage, such as histamine, kinins, prostaglandins, and leukotrienes. So these chemical mediators can also assist in stimulating our pain receptors and will cause an increase in blood vessel permeability. This is vasodilation, and this assists in moving our white blood cells over to the affected area. So here are the steps in how this kind of takes place. We'll dive into this a little bit more in our immune system. But when we have damage, like a splinter going into our skin, we are going to introduce some bacteria into the affected area, and immediately we will have our chemical mediators giving off our chemicals, our leukotrienes, our histamines, and so on. And so we will activate our injured tissues and also our blood vessels that are adjacent to this area. So we'll have a couple things taking place. These chemical mediators will cause these blood vessels to vasodilate, like we mentioned, and this is going to allow our platelets as well as our white blood cells to all come over to the area. Now, this increased blood cells coming to our affected area will cause redness because of the blood moving closer to the surface of the skin. The increase in our permeability, or the vasodilation, is also causing more fluid to come into the area, which is causing swelling, also known as edema. As I said, these cells are moving into the area, and this is changing the osmotic relationship between our blood and tissue. So water is going to follow these proteins by osmosis. So along with the cells, water moving into the area, our proteins being there as well, this is all causing the edema and all the swelling. So we have increased pressure in this area, and that causes pain. It's putting pressure on our free nerve endings that sense for pain. What else do we need to mention? Okay, in step 3, we're also going to have our white blood cells coming to combat these pathogens, the bacteria here. And the ones that are specific to phagocytize here are our neutrophils. Our neutrophils are a type of white blood cells, and when they go through phagocytosis, they might leave some debris behind, and also the cells are going to die themselves here. So they might leave behind some pus as well, that white substance. Let's move into our tissue repair. So our tissue repair, there's 2 different ways of discussing tissue repair. There's regeneration or replacement. Regeneration is going to be when we have our new cells replacing the same type of cells that were destroyed. So if you had some epithelial cells damaged, it's going to replace it with that same exact type of epithelial cell. Whereas replacement, that is going to be a new type of tissue that's going to develop, and usually this produces a scar, and sometimes we have a loss in our tissue function here too. So the way we can find out whether our tissue repair is going to be more of regeneration or replacement is depending on the extent of the wound and also what type of tissue we have there. And we know our tissues are made out of cells. So we have 3 different types of cells, which we've categorized for tissue repair. We have labile, which are going to be capable of mitosis throughout our life. So this would be in areas of our skin, our mucous membranes, our hemopoietic tissue, or lymphatic tissue. We also have stable, which means there is no mitosis after the growth ends, but they can divide after injury. So this is within our liver, our pancreas, and endocrine cells. And lastly, we have permanent. So these are the ones that if they're killed, they're replaced by a different type of cell, which usually leads to scar tissue. So we have limited regenerative ability. So this is the types of cells that we find in our nervous tissue, skeletal tissue, and also our cardiac muscle. There's some promising research being done right now where we can see that if neurons are damaged, as long as that cell body is intact, that nerve has possibility of undergoing mitosis and gaining functionality once again. But once that cell body is damaged, then the rest of the neuron gets destroyed, and we have much more limited circumstances at that point. So there's two different pathways, not pathways, but two different events in which we can have skin repair. There is primary union and secondary union, which we'll get to in a moment. So in primary union, it means that the edges of our wound are close together. So usually these are the ones that are going to heal nicely, and you won't even really be able to tell that there was a wound there at some point. So let's discuss these steps. Do I have another picture here? I do. Let's discuss it here in this tissue repair figure. I thought I had a figure number here for you. This is figure 4.8 in your textbook. So this is an example of mostly primary union. The first thing that's going to happen is your wound will fill with blood, and that blood will begin to clot. That clot contains a protein called fibrin that is assisting in bringing the edges together, starting from the bottom and kind of moving its way up. As this clot dries, we're going to form a scab at the very top surface. And the reason our body does this for us is to prevent infection. So hopefully your neutrophils at this point have destroyed any bacteria that may have entered your skin from the wound itself. And then you can also see here these fibroblasts that are migrating toward the wound site. They're going to assist in producing collagen and repairing the two edges of the skin here. Well, at this point, we're in the dermis. All right, so permeability of our blood vessels are increasing, bringing in blood, our white blood cells. This is resulting in edema or swelling, and our fibrin is now creating a wall around our wound to section off any further infection, basically keeping the pathogens in one specific area so that our neutrophils can phagocytize them and kill the pathogens. Like I mentioned before, our neutrophils are going to leave behind some debris, and also they may die themselves and leave behind these dead cells, and that accumulates and becomes known as a fluid called pus. So fibroblasts, once again, are working to produce collagen. Our capillaries, which isn't pictured too wonderfully here, but capillaries are going to grow into that area to make sure that this area is very well vascularized. And then our fibrin is going to start to break down and become removed. And at this point, we have granulation tissue. So our granulation tissue is very delicate, granular appearing, hence its name, and it's a type of connective tissue that's going to contain all of these fibroblasts that are secreting collagen and elastic fibers. So over a period of time, this granulation tissue will convert into a scar, and our scar is made up of dense, irregular, collagenous connective tissue. The within our secondary union, so I'm going to flip to the screen now. Within our secondary union, the difference here is those edges of our wound are not close together. So we have a greater chance of infection here. And that's why it's recommended that if you do have a larger wound, that you get it sutured up so that it heals better, and also we have a decreased risk of infection. So we have these three circumstances. Our clot may not close the gap. Our inflammatory response is going to be greater, so the patient will have more swelling, more pain, more redness, and oh, went out of that mode. So we said our inflammatory response is going to be greater. Now, with that, we're also going to have a greater amount of granulation tissue that's going to form because we have this larger gap that we need to close on up. And this causes a contraction of our fibroblasts within this granulation tissue, leading to wound contraction. Our wound contraction is going to result in a disfiguring and debilitating scars. Now this of course depends on how large the area is, but if it is a deeper wound within our skin, you might have some accessory structures in here like sweat glands and hair follicles that will not reproduce and function. That's what we mean by having a debilitating scar. You might not have as much movement within that area as well. So that is it for chapter 4. If you have any questions, please let me know, and don't forget that you can comment on these slides on our Google Slide web link that I posted on Blackboard for you. So I hope you have a wonderful break, and I will see you soon.